Good evening. Welcome to the second to last event in the speaker series of the 2021 International Conference on Engineering Synthetic Cells and Organelles. Just to remind you, there will be one more speaker series event next Monday, and then the main conference will take place May the 18th through the 20th. And as another reminder, you can already register for the conference. It's free. Go to the conference website, syncell2021.unm.edu. So today it's a pleasure to introduce first a presentation by Professor HENDRIK DIETZ, who is a professor for biophysics at the Technical University of Munich in the physics department. Professor Dietz completed his undergraduate studies in physics at the LMU, also in Munich. And then he did a postdoctoral fellowship at Harvard Medical School. And after that, since 2009, he has been a professor at the Technical University in Munich. So welcome, HENDRIK.
Thank you, Darko. So I guess I can overrule the screen sharing now, right? I guess I will do that. And I assume you can hear me. Is that correct?
Yes.
Okay, great. Okay, so hello, everybody. Thanks for joining. I'd like to talk a little bit about our work on designing biomolecular devices and machines. First, I want to spend some time explaining sort of the general mission of the lab. So what are we interested in? So in my lab, we'd like to build artificial molecular devices and machines, ideally devices and machines which can accomplish sophisticated functions, maybe similar or even surpassing those known from natural molecular machines. We want to be able to integrate such function modules into higher order systems and then use them for something useful in society. Maybe a question is sort of why am I particularly interested in this particular question? So I'd like to motivate this a little bit with the help of this photo. I think this is a little bit old. This is, I think, probably from 1910-ish or something like this. So what you see here are two horses drawing a carriage, and then there's one of these early automobiles. And it's not clear whether the car is actually taking over the carriage or whether the carriage is taking over the car in that photo. It doesn't matter for the point I'm trying to make. So this is a metaphor. So you kind of can think of sort of the horses embodying the state of molecular biotechnology, sort of everything that we can know to do with molecular devices. Basically, we pick pieces from nature, antibodies, polymerases, molecular motors. We tinker with them a little bit. We can adapt them to a certain extent, but sort of horses remain horses, right? There's only so much that you can do, you know, with a horse. Whereas if you knew how to sort of engineer molecular machines, molecular devices completely from scratch, like we do with cars, right? Then you have, of course, much greater freedom to build something that really suits your needs. And I think we're in an exciting time right now. So we can see that there's a lot of increased control about building increasingly sophisticated molecular devices right now all around the world. I think we're not even there that we could say we have the analogy of a car in terms of synthetic molecular devices, but we will be getting there eventually. And also, I mean, even though like the early cars, they were not so great and people were a long time debating on whether they actually need cars, I think nowadays it's clear that they have completely taken over the role of horses, right? So sorry for this a little bit metaphorical introduction, but with that, I want to tell you what we're actually doing. So we would really love to contribute to this development in some way. And what we're trying to do is to push as much as we can an effort in trying to build complex molecular structures and try to encode them in DNA sequences, nucleic acid sequences. And so basically, without going into too much detail, so thanks to over 30 years of work in this field of DNA nanotechnology, we are in a position nowadays that we can basically sit in our offices and design synthetic molecular structures through a rational engineering approach. And basically the result of that engineering approach is a list of sequences for DNA single strands, which are designed according to a specific set of rules. And then if you actually make those molecules, for example, via solid phase chemical synthesis and put them together in a liquid solution, they will self-assemble into a three-dimensional structure, if you did the design correctly, that is fully specified in the sequences. So one very popular design approach in the field is known as DNA origami. I'd like to illustrate this process with the help of this movie. So this is an illustration. It's not aspiring to be an accurate physical representation of reality. It's just an illustration. So basically what you're seeing here, first of all, is a very long DNA single strand. We usually call this a scaffold strand. It's like kind of the backbone of what's going to be our structure. And then we have these colorful short pieces here. These are short DNA single strands, which we call staples, and they're designed in such a way that they are partially complementary to particular spots on the scaffold strand. So then when you put everything together through the fusion and self-assembly process, all these molecules, they come together and they actually, by Watson base pairing with the scaffold, they fold the scaffold strand into a structure which is fully encoded in sequences, and the sequence in this case of the short oligonucleotides. In this case, you get the product will be a simple rod where you have 10 DNA double helices which are linked together for multiple crossovers into this rod-like shape. Okay, so this was fiction. Now comes reality. So what you're seeing here are, I think, these are 47 objects that we actually made over the course of the last 3 years. And everything that you see, but everything that is yellow and blue actually is made entirely from DNA molecules and sort of completely synthetic. And these objects that you're seeing here, these aren't models or anything like that. This is actual data. So what you're seeing here are structures that we determine of these objects using single particle cryo-electron microscopy. And as you can see, there's a huge diversity of shapes that you can see here, right? And the fact that you can solve the structures using cryo-EM also reflects the fact that these are properly folded and well-defined. So you can build small objects like those shown here in an outer orbit, which may comprise like 50 oligos or something like this, like the small one here. And there can also be gigantic objects like this big yellow ball in the center, which I think, if I remember correctly, comprise about 30,000 oligonucleotides. And then just for basically give you like a sense of scale, there are also two natural objects included here. So on the outer orbit, there's this magenta colored object, which is showing up here in the back. This is a protein membrane pore called YAX-AB. It has a molecular weight of 800 kilodalton. So it's about the same size of some of the other origami objects here. And then this magenta colored sphere here, this is actually an hepatitis B virus core capsid with 30 nanometer diameter. So basically the bottom line is, with the modern methods of DNA nanotech, we are in a position to rationally engineer a huge diversity of shapes on a size range which is commensurate to the scale where natural molecular machines and higher order complexes live. We can also use cryo-electron microscopy to further study these objects in greater detail. So this is one of the example of one of the more recent structures that we have solved, which was run in movie. So this is a cryo-electron microscopy map determined for an object which has no particular function other than to have this weird shape in a way. The point is you can use this structural validation method, and you will actually be able to see every helix in this entire object with a detail that you can recognize major and minor groove. You can see all the crossovers that hold the helices together. And with this level of detail, you can actually go and also refine your designs so that they actually fit specifications. And then with such data, you can also fit and construct pseudo-atomic models. So basically infer the coordinates for all the atoms that make up the structure. And this is something that we did here for this electron density map. So now we have an atomic model for this object. And I think this one comprises on the order of 500,000 atoms. So we have the coordinates for every single one of these 8,064 base pairs where this which this object is made of. Yeah, and I'm rotating it, and then you can go in and analyze the geometry, quantify whether you're happy with particular aspects. You can try to refine the design so that it fits your specifications. And with this kind of process of experimental validation, atomic model fitting, we have now several atomic models available, which we're using also just to troubleshoot our design rules. And I'm gonna just run these movies here, and then you may notice that these two objects here actually look fairly identical. Turns out these are actually two variants of the same object. So the top one is the one I just showed you, and you can see that this object actually has some twist deformations. That's also one of the reasons why we made this. We wanted to study global twist deformations and how they relate to particular design decisions in the interior of the object. So now we saw it's deformed, so we can actually go back to the design and fix that deformation rationally by, repositioning some crossovers. And now we have a new version of this object, which is much straighter. And again, this is then validated experimentally. It's just reflecting the fact that this is really has become some kind of engineering in a way. So then this is a snapshot, basically like somewhere in the interior of one of these objects, just sort of reflecting where we are in terms of the resolution that we can obtain using cryo microscopy. So it's only order of 4 angstrom. That's where we are right now. And this resolution, you can kind of see the discreteness, or like sort of, the discreteness of the DNAW helix. Right, you have these bumps here, which are presumably reflecting the phosphates. Then there's a little depression here. This is presumably a missing phosphate because this is actually the terminus of a strand. Doesn't have a phosphate. And then here, this is a strand crossover. Basically, one of these molecules, changes the helix, and I can rotate it, and you see this little density is actually as a single covalent bond. So I'm actually kind of happy to see this. And now we can, it's at least conceivable that if we can push the resolution a little further, you can actually do rational placement of chemical moieties and additional functionality so that they are at the correct relative positions to, for example, template some catalysis or something like this, but if you want to do something fancy. So then coming back to this overview, you may ask now, what is this good for? All this, so actually there are many answers to this question. So now many colleagues in the field have built various devices for various purposes, starting from sort of applications which are not so material demanding, or where the DNA nanostructures don't need to survive in outlandish environments. So there's lots of useful tools in biophysics and biochemistry for sort of niche science questions, but this is still valuable. And now sort of as the field progresses and we know better how to make more durable structures, more accurate structures, also the applications grow and the sort of in scope. So today I want to share with you something we've been working on for multiple years now. Actually, we started working on this in 2017, I believe. And in 2019, we started working on the type of project I want to explain now. So pre-pandemic. So we're working on as a kind of virus neutralization therapeutic agent. So, and we call this in our lab the Venus virus trap. So for those who are not familiar with Venus traps, so Venus trap is a carnivorous plant that sort of, it can trap insects in a sticky leaf and then digest the insects. So kind of what we want to build is something like that for viruses. And that was an idea that came before the pandemic, but I think it became all the more relevant. And so this is a collaboration, actually. So all the people named here in white are working in my laboratory at TUM. Then we have Werner Kolbe, Florian Wilsch, and Ulrike Protzer. They're from an Institute of Virology, the virologists and physicians, also located in Munich, German Center for Infection Research. And then we also have help and a lot of input from soft matter physicists from Brandeis University, notably Mike Hagen and Seth Fraden, who also was in the lab for a year. And then these are the people who have been driving, who drove this work in my laboratory, so Christian Siegel, Anela Wilner, and then 2 postdocs, Wouter Engel and Jessica Kretzmann. And I think they did a great job. So, okay, again, what is the goal of this work? We want to build a macromolecular device that can trap viruses. And the idea is that if we can trap viruses and engulf them completely, we can prevent potentially viruses from undergoing interaction with host cells. And sort of inspired by neutralizing antibodies, if you could do this, this would have the potential to reduce the viral load in acute infections and just give the patients more time in developing an effective immune response to get rid of the virus. And I'll say more about that therapeutic window at the end of my presentation. So what I'm going to tell you now, how this basically builds some previous work. We made some important progress in building large structures in 2015 and 2017, which enabled us to build the kind of objects I'm going to share with you now, which are not yet published actually. So, so basically what we're building is we're trying to build pretty large, massive, pretty large macromolecular shells that are even bigger than actual viral pathogens. So like 100 nanometers or even bigger. So the approach that we chose is as follows. So we're building triangular building blocks from DNA. And what you're seeing here is a cryo-EM map, actually, one of these objects that we built. So it's an equilateral triangle. And the edges are beveled at a particular angle. The angle is chosen such that you can take 20 copies of this triangle, and when you put them together by the edges, they will form a closed icosahedron. And then also, if you look at the edges, you will find that there is some funny protrusion here and then some recesses, and they are actually shape complementary. They kind of encode a particular relative orientation in which you can click copies of these triangles together in order to form such a shell. And then here's another variant of such a triangle that we've built. It's an isosceles triangle, so this edge is actually longer than the other two. And this one also has a different rule set encoded in terms of how copies of this triangle will interact with each other. For example, here's one protrusion, and this protrusion fits only into a hole in this edge. And then here's a protrusion on a bottom edge, which actually fits into the same edge. So this is a particular rule set in which this thing can polymerize. Also, the bevel angle here is more shallow, which allows this triangle to oligomerize into a bigger shell. So with these kind of tricks and using sort of careful engineering, basically driven by data from cryo-EM, we were able to make variants of these triangles, which self-assemble quite efficiently into these higher order shells. So you can make octahedra, icosahedra, and then this shell, which actually contains 60 copies of this triangle. And then this shell actually contains 80 copies of 2 triangles. So, so this actually approaches, well, this one, no, we have an even larger shell, which is not on the slide because we don't have a cryo-EM structure, but even larger one actually has 180 triangles. That's about 1 gigadalton in molecular weight. So it's pretty big. So then this is pretty robust. So you can take these shells, for example, and just stick something on it, and they will still assemble into nice icosahedrons. So we made a spiky version of this icosahedron just to solve the cryo map, just to see whether that would actually work, and it did. So then here's a raw EM micrograph just to see how that actually looks. Sort of, we just look at single particles. So this is the T=3 capsids, many copies of them. They look a little bit like clean viruses, sort of, and then here is maybe some of the building blocks. But overall, the yield is actually pretty good, thanks to the refinement that we did with the structures. So then, yeah, I can skip this. So then, this is a cross section of the icosahedral shell. So you see this has this thick wall and all these circular cross sections. These are affecting the cross sections of individual DNA double helices. So this one has a diameter of 100 nanometers. The cavity is about 80 nanometer in diameter. This is the bigger shell made from 60 of these triangles. This one has a cavity of 150 nanometer. So you can actually stick, if you wanted to, you could stick the icosahedron inside. And why I'm putting the icosahedron in here is because this is pretty much identical to the average dimensions of the influenza virus, or like even the SARS-CoV-2 virus. Actually, it would fit into the shell. So they have achieved the appropriate size with these shell designs. So now, which raises the next question: how do we actually trap viral particles in these shells? So basically, there are two different routes that you could follow. You make shells which actually pretty much like a carnivore's plan. It's basically is ready, and it's patrolling through the body fluid, and when it viruses can enter through some aperture and then they get stuck there. There's one route. The other route is that you kind of dynamically assemble these shells around viruses to fully engulf them. We have invested most of our work actually in this avenue, and to do that we had to learn how to make partial shells which are actually not fully closed, right? Because you need an aperture so that the viral particles can still diffuse in. So I'm sharing with you some variants. So this is a variant of the icosahedron shell, which we re-engineered so that basically the multimerization stops at a half icosahedron. So, and again, it's a well-defined structure, assembles with decent yield so that we can actually solve a cryo map of it. And it's like a half icosahedron. So now you could imagine also you could have two copies of this half icosahedron, they could click together, right? And then here's another variant, which is a little bit more close. Now, basically one of the pentagon vertices of the icosahedron is missing. So this has a bigger, it's like more like a bowl. So it's a bit more floppy here, the edges. That's why kind of this looks frayed, but sort of it seems to be well formed. Okay, so now we have shells with apertures. Now we need to figure out how we can actually trap viruses. How can we make the shell sticky for target virus? So I want to illustrate the many options that you have there with the scheme here. So imagine you have one of these shells. So since it's made of DNA, we know where all the DNA strands are. We can site-specifically conjugate, all kind of moieties to the interior surface of these shells. So one moiety that comes to mind is antibodies. There's plenty of antibodies available against all kind of stuff. So what you could do is you covalently link a DNA tag onto these antibodies, and then you could make a variant, for example, where the entire inner surface is completely plastered over with antibodies. That's conceivable. But you can also put their host receptor domains. An example is, the ACE2 receptor on the lung cells, for which the spike-2 protein is sticky. You could take this receptor domain and just present it here as a kind of a decoy to trap the SARS-CoV-2 viruses there. There's peptides, aptamers, many options that you could work on. So just to prove a concept, we made some variants of these shells to which we conjugated antibodies in the first set of experiments for an hepatitis B virus core capsid. So now we have these are half octahedra, and you can see here some of these spherical objects in there. These are actually hepatitis B core capsids, which are specifically trapped by this half octahedra by antibodies, which we conjugated in the interior surface of these octahedra. You can also do this with the bigger shells, and then get pictures like this in an electron microscope. Again, these spherical particles, these are hepatitis B virus core capsids. And then here are our DNA shells, and they're specifically functionalized with antibody against this hepatitis B core capsids, and you can trap them in there. Okay, so this works, and here you have many the viruses are here in excess, so that's why not all of them are sequestered actually in these shells. We solved a cryo map actually of a hepatitis B core capsid trapped by two half octahedra. So in a particular stoichiometry, you get many of these structures actually. It looks kind of funny. It looks like a TIE interceptor from Star Wars. So the little sphere here is actually hepatitis B core capsid, and then you have these two DNA shells which coordinate this thing. And then if you increase the density threshold, and then you can actually see there's a signal in between the virus and the wall from the DNA, and these are sort of randomly fluctuating antibodies which hold the viral capsid inside. Okay, now next question that we have is, does the virus trap actually prevent virus surface interactions? We did plenty of experiments to this extent. Looking at the time, I'm going to skip one and just advance. So we did much work in vitro, so to speak, with sort of artificial surfaces to troubleshoot the surface blocking capabilities. And now I'm going to show you some work with live cells. So what we did is the following. We have here a pristine human cell line. And what we did is we took adeno-associated viruses, as a model system for an infectious virus, which were carrying an EGFB gene, green fluorescent protein gene. So here the idea is if such a HEK cell is infected, actually, it will express green fluorescent protein, which you can evaluate then using flow cytometry or fluorescence microscopy, for example. And then the idea is now we can sort of, we have a known amount of viruses which we give to cell culture. We could add our shells, for example, specifically modified to trap the adeno-associated virus. Then we can evaluate how many of these cells are actually still turning green, right? That's the way how you would basically measure neutralization. So then this is how this looks like. So this is sort of a first microscopy view of many cells. So here's the nuclei stained with a different dye. This is the EGFB channel, many cells turning green when you just give them a certain at a certain multiplicity of infection, adeno-associated viruses. And by comparison, if you add these shells, then actually very few cells turn green. So the shells actually successfully prevented the viruses from entering the cells. There's still lots of cells here. You can see the nuclei channel. And now an important control experiment is sort of to just do the same thing with just the antibodies at the exact same concentration, effective concentration at which we had antibodies here. If you do this, we still have many green cells, which shows you one key advantage of these shells. Basically, they allow multivalent binding of these antibodies. And this is much stronger than if you have individual on and off binding of these antibodies, right? Here you need many more antibodies to actually effectively, completely passivate the pathogen surface. All right, so the cells were happy, just skip this. And then the latest news is that we also did this with actual hepatitis B viruses in human cell lines. And this is quantified in this way. You see here sort of the neutralization is measured by quantifying the amount of a viral antigen that is shedded by infected cells. So you can do this, for example, 4 days post-infection or 8 days post-infection. And we have shells which are specifically functionalized with an antibody. And if you only give the antibody exact same concentration, you basically get zero neutralization. All the cells that get infected with the hepatitis B, all of the concentrations we looked at. But if we put the antibodies in our shells, get very, very efficient neutralization. This is all very good and promising. So the virus trap concept works with live cells in cell culture and nasty physiological fluids with nucleases, etc. So now a question, which is more a little bit of an outlook, is does this actually have any therapeutical merit? So this is something that we need to answer in the future. I just give you some few interesting points. So you may have heard about monoclonal antibody therapies as a therapeutic intervention, specifically for COVID-19. So there is the FDA actually has granted emergency use authorization for this REGEN-COV. It's an antibody therapy. It's a mixture of 2 antibodies which are neutralizing the SARS-CoV-2 viruses. President Trump got some of these when they were not yet released. And the idea is that they do exactly the same thing. You add the antibodies into circulation, they neutralize and prevent the viruses from interaction with host cells. Similar ideas also pursued with these designed mini protein inhibitors. So I think there is a therapeutic window here potentially. We don't know yet how this stuff will work in an organism, something that we need to test. And a good question you might ask is why not just use antibodies in the first place? Well, there are many answers to this question. First of all, you could, but in many instances, neutralizing antibodies may not be available. And they're also subject to mutational drift. So they need to bind in a very specific way on the viral surface in order to be neutralizing. And it's actually hard to get. And with the shells, you don't need that. And you can think of these shells as a sort of really structural scaffold that can enhance antibody efficacy through avidity, right? Even if the antibodies are individually weakly binding for multivalency, they will bind strongly. And then you have the occluding effect from the shell material. And then there's a little subtlety. Another advantage is that you can basically you can use any virus binder and multiple virus binder and different virus binders to just specifically target a viral pathogen. So we're excited about this. So we'll continue to push this. Going to test this on organisms in mouse. One option is also to look into Omnivore virus traps, something that can sequester different types of pathogens. You can think of influenza and SARS-CoV-2 and maybe something else. And then we also have the option to include virucidal moieties. You can think of proteases, for example, enzymes which will destroy surface proteins on the viral pathogen and really deactivate it. It's another interesting direction. So there's a consortium that we have formed. It's funded by the European Union. I have the honor to lead this. Includes virologists, physicians, chemists, and us, nanoengineers, as we call ourselves. And then hopefully we can bring this to a sort of level of technical readiness that we can make an informed decision about whether this is sort of something worthwhile to pursue for in the future. Okay, how's the time, Darko? Darko, you're muted. I'm sorry.
Take a few more minutes if you'd like.
Okay, so I have two more things that I can advertise. I cannot talk about this in detail, but both things that I'm going to share with you briefly, they're all posted on bioRxiv. You can look at the complete survey you want. So two more things. So one interesting study led by Eva Bertolzin in my lab, really talented PhD student, is a nanoscale reciprocating rotary mechanism with coordinated mobility control. That sounds fancy and maybe a little bit hard to digest. I want to tell you what that means. So it's actually, by the way, it's a work in collaboration with Alexei Aximentiev lab and Christophe Maffeo at University of Illinois at Urbana-Champaign. So what we built is a rotary mechanism in which, much like an ATP synthase, basically the rotation of a rotor induces cyclical reciprocating motions in the stator. Basically the orientation, where the rotor is, it will deform the stator in a particular way. And this is kind of how you can get basic coordinated motion. So what you see here is a movie from a simulation that was done in Alexei's lab, where basically the rotor was forced to rotate in a particular way, and you can see how the structure basically cyclically deforms. So basically, much like an ATP synthase, where the gamma shaft, as it's rotating through the different state orientations, it basically pushes the stator pieces outwards. So with this mechanism, at least you have a coupling now, where the orientation of it can induce specific, deformations in the stator, and then we hope that we can exploit this in the future to actually drive rotation through chemical reactions. Or maybe if you mechanically rotate this shaft, we could maybe use this to do chemical synthesis uphill. And Eva actually did a lot of single molecule experiments with using single molecule fluorescence and also single particle cryo-electron microscopy to verify that this mechanism actually works. And you just basically just more like an advertisement. This very few example structures. This one is the most interesting one, basically taken from an analysis of heterogeneity in cryo-EM data. We can reconstruct basically a movie of a moving rotor inside of a stator. And the other thing that I want to share with you, also posted on bioRxiv, is this study. It's a synthetic tubular molecular transport system. So we asked ourselves the question whether it would be possible to generate a one-dimensional random walker from DNA components, which is as fast as a natural molecular random walker, like, for example, kinesin motors in the absence of ATP. And we succeeded in doing this. Pierre Stummer worked on this, again, a very talented PhD student in my lab. And here's a movie of a DNA random walker labeled in green. This movie is approximately real time, running on a DNA filament labeled in red. This is about 3 micrometer long, and basically it's moving. I think this system is about 500,000 times faster than previous artificial 1D random walkers. And if you're interested in how we made that happen, please have a look at the paper. And maybe we can publish this eventually in some actual journal, but it's on bioRxiv, so I have peace of mind. So this, I want to thank you for listening. Again, I have my little metaphor here of the cars and horses, which I love at this moment. If you want to get in touch with me, here's my Twitter handle, so you can follow me on Twitter. You will be updated with our latest publications. There's our website if you want to check it out. My email address, and then here I want to highlight all the members of the lab, current scientific staff, everyone working really hard to make the whole machine work. Now I take any questions. Thank you for listening.
Thank you very much, Hendrik, for a fantastic, inspiring talk and beautiful microscopy pictures. It's so exciting motors and walkers. But we have a couple of questions about the virus trap. So one question is, are the DNA shells stable in actual physiological conditions?
Yes. Yes, that just took us about a half a year, but the answer is yes, because I mean, all of this would be completely useless if they weren't stable. So the answer is yes, there is a particular treatment we do that makes them stable.
Thank you. And related to that, so after the trap traps the virus, will it be degraded in the body, or does it just float around as a neutralized virus?
That's a good question. We don't know the answer to the question. We assume it will get degraded. And my virologist partner in crime, Ulrike Potzer, says, even though, I mean, you could hypothetically, you could think of the following scenario: you have the virus trap, it sequesters the virus. After a while, the shells get degraded, and the virus is free again to do its job. Basically, that's the scenario. But our virologist says that actually anything that you do to somehow delay to somehow reduce the infectivity will help buys the body time to mount a proper immune response and kill off the pathogen. So this is even if that would be the case, that could be useful. But we have lots of additional ideas, right? I mentioned we could include proteases that would degrade viral surface proteins, and then the virus would be broken and could no longer do its job.
Thank you. We have one more question, a comment that says the venous virus trap is a beautiful tour de force. And are there extensions of this approach that could be applied to antibiotic-resistant microbes?
Yeah, that's a good question. I think the issue with microbes is that they're here. So this idea of engulfing them will be hard to do because they're so much bigger, obviously. So you would have to do something else. So a while back, I was very much excited of building artificial membrane pores. So kind of devices that could sort of punch pores into membranes. And the idea was that maybe we could build such kind of pore punchers so that they specifically lyse bacteria. But this, I think that's something for the future. But I don't think that shells will do the job. There's too much material that you need.
A few more questions. So the work that you describe focuses on achieving function through elegant engineering of structure. How about extending into dynamics to engineer biomimetic nanobots, such as artificial mimics of motor proteins? Are you planning to systematically explore design rules for such potential nanobots for applications such as in harsh environments?
All right, that's a good question. So, yeah, I mean, we're working on trying to actually establish actual motors. So the two mechanisms that I shown, basically these are passive mechanisms that have degrees of freedom, but they're just randomly driven through these degrees of freedom through thermal forces, right? There's no active process that would actually somehow rectify their motion. So we're working on trying to establish ways to make actual motors. And if we had motors, then we could potentially also nanobots that do work in whatever the environment is. But this is something for the future.
Okay, I'll ask one more question before we move on, and that is, are the DNA nanostructures immunogenic?
It's also a very important question. So I think the answer to this question is not yet clear. So we have been working on studying the innate immune response to these kind of structures, and there is basically two kind of mechanisms that are part of the innate immune response. So there's these toll-like receptors that can detect CG motifs in DNA on the extracellular side. And if you could imagine just making structures which don't have those motifs. CG-free, so you can basically bypass that detection method. And the other part of the innate immune response is an intracellular DNA detection method in the cytosol. Is something that the cell considers as potentially harmful. This could reflect intrusion by a virus. So there's enzymes and proteins that detect DNA intracellularly, and we're in the process of studying that response. And I cannot give you like a clear answer at this point. So to some extent, they might be immunogenic, but this is something that needs to be studied.
Thank you very much. And thank you very much for your talk.
Thank you for listening.